I welcome you again to the Wednesday seminar, and I'm gladly going to present to today's speaker, which is Dr. Anna Davies-Barrett, who is a research associate in human paleopathology on the UKRI-funded project Tobacco Health and History. She works as an osteoarchaeologist, specializing in the analysis and curation of human remains, with a focus on the study of ancient diseases. She has an interest in respiratory conditions, including the effect of air pollution, environment, living conditions, and cultural behaviors on respiratory health in different past societies. Prior to coming to Leicester, Anna worked as a lecturer in human osteology at Cardiff University and as a bioarchaeologist at the British Museum, where she investigated respiratory diseases in populations from ancient Sudan. And once again, welcome to our speaker.
Thank you. That was a nice intro. All right, so I have to stay around here because the camera fatigue.
No, we have a new camera
I can wander around.
Yeah.
Great, because I always do. Can everyone hear okay? Okay, great. Well, thanks for giving up your Valentine's Day to be here for me. I'm so honoured. I'm going to be talking today about the Tobacco Health and History project. So for anyone who doesn't know, this is a project that's been based at Leicester for the last 3 years. We're just gone into our 4th year now. And basically, we are investigating past impacts of tobacco consumption using osteoarchaeological and biomolecular approaches. So just a bit about tobacco to start off. So tobacco is not native to Europe. It was imported from the Americas around the 16th century. That's when it first was imported. And then it started to be cultivated in Europe really quickly. By about the mid 16th century, it became very popular very quickly. And originally, it was actually a really strong and powerful intoxicant. It could cause hallucinations, it could cause fits, it could even cause death originally. But as it was adopted in Europe, it was selectively bred to be more and more addictive, but less and less kind of physically impactful on the body. And as with other popular food commodities during this period, it's linked to mass importation, colonialist practices, and exploitation. So there has been a lot of historical work on these aspects of tobacco, not so much on the impacts of tobacco on health when it was brought into Europe. I'll get into that a bit more in a second. So tobacco in the 18th and 19th centuries, this is where the project has done work in the Netherlands and Britain. Today, I'm just going to focus on Britain because it's just a bit easier. I don't have to explain 2 different historical contexts, all that stuff. But in Britain, there were huge quantities of tobacco consumed across the country. At one point, I think around the 1700s, there was enough being imported into the country, let alone being grown in the country, to provide at least 1 pipeful of tobacco for every person in the country. So it's being consumed in massive amounts. And it really becomes very quickly integrated into British culture. It becomes part of everyday life, and it's the thing that's the most favourite, kind of it lasts throughout the time, is pipe smoking. So that's gone out of fashion now, but for a really long time, pipe smoking was kind of the major way to consume tobacco. But snuff rose in fashion later on. So that's powdered tobacco that you take up the nose. It's quite disgusting, but at one point it was considered very fashionable, very smart to be taking snuff instead of smoking dirty pipes. Chewing tobacco was also popular, but not so much in Britain, mostly over in North America. And then in the 19th century, we see a rise in popularity of cigars, and in the late 19th century, cigarettes. So this is all based on rises in fashions and political allegiances as well. With France came snuff, and then as political allegiances with Spain changed a bit, cigars came over from there. So historical perceptions of tobacco, what we understand about consumption of tobacco in the past, comes mainly from historical sources. And these paint the picture that smoking was predominantly a male activity. It's done around social activities, it's done around being with your mates in the pub, pretty much the same as how it was about 20 years ago or so. It's all about drinking. It's all about being with your friends and having a pipe and having a pint. And often in the taverns, you could buy a pint and a pipe full of tobacco straight from the counter. Women who smoked were painted as poor labourers, prostitutes, old or really odd, and drunks. So smoking was really seen as very undesirable in women from what's being kind of written about historically. And we also see that the manner of tobacco consumption is linked with class and socioeconomic status and fashions at the time. So again, the cigar rises in fashion, snuff rises and falls in fashion, and snuffing was seen as more womanly. It was okay. It was you know, more acceptable for a woman to snuff than smoke. So, and I'll come back to all of that. That's all important, but I'll come back to that in a bit. So sorry for all the smokers in the room. I always have to give an obligatory apology because I hate shoving it in your faces, but we all know that smoking is really bad for your health. You know, it's no surprise. It damages the lungs, it increases susceptibility to respiratory infections, including chronic obstructive pulmonary disease, asthma. It increases instances of tuberculosis, pneumonia, all this kind of stuff. It damages your heart and circulation, increases risk of cancer, causes systemic inflammation, suppresses vitamin metabolism, and promotes dental oral diseases, and loads more. And actually, a lot of the ones listed here, we can look at in human skeletons. So we can actually investigate what is going on in the past in terms of diseases and whether that correlates with smoking. So no one's actually really done a lot on this. There's been a few osteoarchaeological papers that have looked at tobacco use on health in the past, but very, very few. A couple on dental disease that have shown higher prevalences in smokers, one or two on respiratory disease, but really not much is done. And this is really interesting because the number of clinical studies done today on how bad smoking is for you, it's really, we all know it's terrible, but there's not a lot written about it in the historical texts or texts talking about what was going on in those periods. There's just not that much done, which is crazy because it's a really interesting avenue of research. So on our project that Sarah in Skip, for anyone who knows, put together, she really wants to know what were the long-term consequences of mass tobacco use within a population. So how, when we suddenly have this massive influx of tobacco coming into the country, how is that changing what diseases look like, how people, you know, what kind of diseases people are getting, are they becoming sicker more often, are they getting more chronic diseases, that kind of thing. And we can look at that in the archaeological record. It's not easy, but we can. And also, can we learn more about who is smoking in the past? So how smoking relates to your status, gender, age, all kinds of things. And does what we're seeing fitting with the historical perceptions of who was smoking? And how did widespread adoption of tobacco affect health across time? So kind of all the stuff I've just pointed out, but from an intersectional perspective. So are there different health impacts in different social groups? So if a very high class white male from London was smoking and a very poor class black female from London was smoking, are we going to see differences in how those two groups of people are going to be suffering from diseases? Because one might have many other buffers that stop them from developing diseases that the other one doesn't. So it's not just that they're both smoking, but also all the other factors that affect life that can change the outcome, the disease outcome. So how do we determine who was smoking archaeologically? So osteoarchaeological methods are based around looking for two things in the human skeleton, and that is one, pipe notches, so that's the top corner, and these pipe notches are caused by abrasion by a specific type of clay pipe. So this pipe here is called the cutty pipe. It's quite short and it has a clay stem, and the clay from the stem quite abrades the dentition to cause this distinctive circular hole. So we call this a pipe notch. What we also see is dental staining. So You might have seen in very chronic smokers stained teeth, brown stained teeth. We see that in archaeological skeletal remains as well. We see them on the lingual, the inner surfaces of the teeth. We tend to see a brown kind of staining pattern. And this is useful because we can say, okay, these people definitely smoked. But in some ways, it's not quite so useful because other forms of tobacco consumption are unidentifiable. So if someone chewed tobacco, we're not quite sure if it causes this staining, but it's not, yeah, we're not determined whether or not it causes staining. So we can't say for certain that people were chewing tobacco. We can't say if they were snuffing tobacco. And if they don't smoke at such a degree that it causes staining or pipe notches, so they might have just smoked a little bit every now and then, we also can't tell from the skeleton. So this is kind of a bit of an issue for us. And one of the other main issues is that if we don't have the dentition, we can't tell who smokers were. So it's fine. So for the project, we've kind of gone, okay, we've got some skeletons that have a complete set of dentition and they've got no evidence for smoking, so they're probably non-tobacco users. We can't say for certain, but they're probably non-tobacco users. And then we have the skeletons that have pipe notches, and we can say, okay, and staining, and we can say, okay, they were most likely consuming tobacco. But we have this massive group in the middle of individuals who've lost their teeth, not necessarily their entire set of dentition, but even just parts of it. And if we're missing just a chunk, we can't say whether or not they were smoked or not, because there might have been pipe notch or lingual staining in the teeth that are missing. Does that make sense? So we have this massive shortfall, particularly in older individuals, because particularly in the post-medieval period where dentition, it was just, dental health was awful. So, you know, you didn't have to be that old before your teeth started falling out all over the place. So in older adults and populations from sites where the dentition is not very well preserved, we don't know who was smoking and who wasn't. And this is kind of a massive shortfall for us in terms of getting enough individuals to actually look in detail at how disease prevalences were changing and whether different groups are affected. Question mark, what do we do with these people? So Sarah, who is the ultimate genius, she heard about this approach called metabolomics. So metabolomics is the study of small molecules called metabolites that are produced and transformed within an organism. So anytime any kind of physiological process occurs in your body, certain molecules are produced, these certain small metabolites are produced, and these are specific to the physiological process occurring. So say, for example, you smoked tobacco, that's going to cause a reaction in your body that's going to produce very specific metabolites. And if we look for those very specific metabolites, we can say this person has these metabolites, they were a smoker, this person doesn't have these metabolites, they weren't a smoker. And this is used in medical research very extensively, and all kinds of other fields of research. It actually gives information, so not just tobacco use, but all different kinds of processes create specific metabolomic signatures. So we can look at genetic and epigenetic factors, aging, environment, different diseases, drugs, your nutrition, lifestyle. So this is all used in medical practice to help investigate some processes occurring in the body. So Sarah thought, well, can't we apply this to human bone, archaeological human bone, and see if we can identify any small metabolites in the bone that could give us specific signatures to tell us about the physiological processes occurring, specifically with tobacco consumption. So that's what we decided to do. We took very small samples of human bone. We managed to get down to 50 milligrams. So for anyone who's a bit destructive analysis, which I am, I don't like hacking into human bone. This is a really minimally destructive approach, so it's really great. So we take this small sample from all these individuals that we want to look at, and we run it through a metabolomic workflow, which includes using mass spectrometry. And it's quite complex, so I'm not going to go into it in huge detail, but at the end, what we get is the detection of molecules specifically altered by tobacco consumption. So this will help us identify tobacco users. So we were like, okay, in theory, let's apply it, let's see what happens. And we took 2 populations, one from Barton upon Humber near Hull, 154 individuals, very rural, mainly agricultural population. And we took another sample from St James Garden Burial Ground, Euston, London. So this was one of the massive cemetery that was excavated during the HS2 excavations. If anyone knows about that, it was on the BBC and all sorts. And this is a very urban site with lots of different occupations, and we have, really importantly, different status groups, 4 different status groups for this population. I assessed them all for age, sex, pathology, and evidence, osteoarchaeological evidence for tobacco use. And then we took samples from them. And this is what we got. So these results are way better than we were expecting them to be. They really are. So these individuals are the ones where osteoarchaeologically I went and said, yes, that's probably a smoker, and yes, that's probably a non-smoker. And so we ran them through with the metabolomics and did some stats, did some data stuff with them. And what we got at the end is a very clear separation according to their metabolomic signatures for smokers or tobacco consumers in blue, and to non-tobacco consumers, so people who probably didn't consume tobacco, in orange. This first one is for Barton upon Humber. You see a really nice difference between the 2 groups. And then we also have the one for London, and you can see that it's slightly more mixed. And we think this might be because of the influence of a polluted environment, and possible secondhand smoke is altering the metabolomic signature in a similar way that tobacco might have done. So you get a mix of non-smokers and smokers together. So looking at this, we can then take out individuals where we don't know whether they're smokers or non-smokers, and we can plot them statistically in against these groups. And we can say, okay, this person falls within this signature, they're a smoker. This person falls within this signature, they're a non-smoker. And this is really exciting because we can now say whether individuals were in the past were smokers or non-smokers without having to use osteological methods. So here's the plot of the individuals I was able to tell osteoarchaeologically who were smokers or not. We can see at Barton upon Humber, it's a very small amount. There's a huge number of individuals who we couldn't tell whether they consumed tobacco or not. Similarly at London, it's all down to preservation and older age groups, all that kind of thing. But, oh yeah, you see, big chunk from Barton upon Humber. Once we apply biomolecular methods and add those individuals in, the number of individuals who we don't know whether they smoked or not is shrunk right down, which is amazing from us because it gives us a much greater sample size to do analysis of disease and any statistical tests. And just to show how really useful this is, these are our age categories from young adult, middle adult, old adult. Older adults who have lost a lot of their dentition, it's really impossible to tell with most of them whether they smoked or not. And this age group generally gets overlooked in osteoarchaeology quite a lot because of issues like this. And we have just completely shrunk the number of unknowns in that age group. So we have a much larger sample size for that age group, and we can then look at the impacts of disease in that age group much more. So it's really important for us to be able to do this. So having done all that, what did we find out as part of the project? What are our results? So one of our questions was, who is consuming? So what parts of society were consuming tobacco? And although historical texts say that it was really undesirable for women to smoke, and only the poorest and lowliest of women, only the prostitutes, the labourers smoked, actually what we can see is that it's pretty even, particularly at Barton upon Humber. Similar rates of men and women were smoking. In London, there's a little bit less women, but there's still almost 50% of women were smoking or consuming tobacco. So this is really interesting because it directly contradicts the historical text and what they've been saying in there. And when we look at just, so this is with the metabolomics added in. This is just osteoarchaeologically. So when we look at osteoarchaeologically, this gives us depending on whether there's a pipe notch or just staining, this might give us a slight indication as to how people are consuming tobacco. Because if you have staining but you don't have a pipe notch, you might have been using certain types of pipes that didn't cause a pipe notch. You might have been smoking cigarettes or cigars. You might have been consuming in a different way. You might have been really careful not to make sure that you're not developing these notches. So we can see here are the males from both sides, and we can see that pipe notches are pretty prevalent, and lingual staining is fairly prevalent, not as prevalent. In the women, we just don't get pipe notches. We get loads of lingual staining, but we just don't get pipe notches. The reason this is really important, and that we found these results, is because the very few archaeological studies that have been done already using osteoarchaeological methods have not looked at lingual staining. They've only looked at pipe notches. So they are agreeing with the historical texts that say that women weren't smoking because they're not seeing pipe notches in women. But looking at the lingual staining, looking at the biomolecular methods, we can definitely say that women were smoking, and they were smoking at a similar rate to men. So this completely changes our historical understanding of who and who wasn't smoking. Also, looking at status, we had some kind of surprising results. We thought maybe high status women wouldn't be smoking as much, and low status individuals would be smoking a lot more. Maybe there's some biases around cultural perceptions of smoking today impacting that as well. But we can see that it does vary between different status groups. We do have a dip in the middle high status females. So perhaps for them who were kind of trying to be a bit socially mobile or paint a certain picture of themselves, women weren't smoking as much because of those negative connotations of smoking in women. But we still see it fairly high in high status, and actually one of the only women we found a pipe notch in was from the highest cemetery site, burial ground in London. So she was very affluent. She didn't seem to care. She had a pipe notch. It's great. And then we see a peak in middle low status males. These are kind of manual labourers, these kind of people. And and actually lower status gets a bit lower, but maybe that's because people just this is a pauper cemetery, so these people were really the poorest of the poor. They couldn't afford their own funerals, and maybe they just couldn't afford tobacco in the same way the other groups could, and didn't consume as much. So this is really interesting. Then when we look at males and status groups, because we don't have pipe notches for women, so I've excluded women from this graph, but when we look at the type of consumption with males, we see that high status males were not forming pipe notches, so they were consuming in a manner that did not form pipe notches. Whereas when you go down to the middle and low status groups, pipe notches are much more prevalent, and in the low status group, almost all of them have pipe notches. So this is really interesting in terms of how obviously the pipe notch is a very visible thing on the teeth. Like you can really see that when you say if you smiled, you can see a big hole in the teeth, right? So people will know what kind of smoking you're doing. So is there a conscious choice by certain social groups to avoid forming those notches? So going into this a bit further, historical sources are only telling us part of the story, and they kind of it's a bit of a mislead about the way people are smoking in society. Like women of most classes were consuming almost as frequently as men. It's just that the way women consuming was different. So here, I love this image because it kind of sums up exactly. Women were smoking in private. They weren't going out to taverns and smoking. They were making sure maybe that they're not forming pipe notches, so they're not obvious smokers, but they were certainly doing it. I like this man kind of peeking in, spying on what the women are doing. Whereas, so the people who are writing the historical text are mainly men, and they're probably not so concerned with like the private sphere of women. So we're not seeing any reference to women smoking, but there are some historians who have started looking and trying to change that narrative, and they have been looking at women's traveling diaries, private written texts from women, and we can actually see that tobacco consumption was occurring, but it's really not in the mainstream thought. So in the texts that are written today, like books on historical perceptions of smoking, they're mostly written by men, and they are not talking about women smoking. It's all about, oh, this activity that men were doing in the past. So that kind of needs to change, and our results are really helping with that, I think. And we can also see that pipe notches, men of different classes were consuming differently. So they cared about being, high status men cared about being seen with pipe notches. And it might be because pipe notches are caused by the cutty pipe. So the short stem pipe. Here you can see one man smoking a cutty pipe, one man smoking a longer stemmed pipe. And the longer stem pipes have to be held like this, and they're more for leisurely, they're more related more to high status. You can sit around, you can hold your pipe, you can smoke, you're not doing anything. The cutty pipe can be clenched between the teeth. So you can be working all day and smoking your pipe. And so this association of the cutty pipe being clenched between the teeth is probably becoming associated with labouring status. So men of less high status, men who are working with their hands all day and holding a pipe between their teeth. So these high status men don't want to be seen as labourers. They don't want to form pipe notches. We also see that there become, this is just kind of an extension of what I just spoke about, but we can also see this image here is a caricature. You see these horrible derogatory caricatures in the Victorian period, and here we have one of an Irish man. So hopefully everyone knows kind of this contention between Victorian English and the Irish at the time. And you can see like bottles scattered around and indicating he's a drunk. He's got a pot on his head, so he must be an idiot, I guess, and he's smoking a cutty pipe. And what we find is that ethnicity really becomes interlinked with the ways people are depicted as smoking. So the cutty pipe really becomes connected with Irish people, and poor labourers, drunks, prostitutes. This pipe is becoming associated with those, and becoming interwoven with all these derogatory ideas about ethnicity or class, all this kind of thing. So the cutty, depicting people using the cutty, kind of becomes a form of marginalisation. So we've got two black women here who are smoking a cutty pipe, both kind of derogatory characters. This one at the top is supposed to be a prostitute, according to the title of the thing. So we really see that this the depiction of the cutty is being used in a kind of almost a political way. But the cutty pipe was also kind of taken back by the Irish, who decided, although they were being depicted with it and it was being used in a derogatory manner, they decided to start stamping their cutty pipes with kind of home rule slogans and political statements and that kind of thing. So they kind of took it back for themselves and became quite proud cutty pipe smokers. And I'll just go back a slide. So yes, we've got predominance of historical commentary for smoking as a male activity. Smoking in women is seen as unfeminine or undesirable, but we actually see that this is probably an attempt by almost exclusively male commentators to shape and control what they perceived of as a male activity. So this resulted in perceptions of smoking as an activity undertaken by and of greatest importance to men, and this persists in our knowledge today of this period, and it's not. Our project has shown that that is not true. Okay, that's one side of it, who was smoking and why. Now I'm going to talk about the health impact. So this is kind of my specialism, paleopathology. We haven't done huge amounts on the disease data yet. There's just so much of it. So I'm just going to give you kind of a small case studies overview. So when you have inflammation on the skeleton, in regions of soft tissue near the skeleton, you get bone reaction. So you can have new bone growth on the skeleton in regions where inflammation is. So that's what I look for to identify areas that were inflamed in people when they were living. So one of those is maxillary sinusitis, is your sinuses here. And sinusitis is literally, it just means inflammation of the sinus. So I look for new bone forming within the sinuses to see if people had inflammation. And obviously, respiratory disease, smoking, you'd expect smokers to have higher rates of sinusitis in the past. And what we found is actually in London, pretty similar rates. At Barton-upon-Humber, we do have a slight increase in smokers. The odds ratio for that was 2.5. You were 2.5 times more likely to have maxillary sinusitis from Barton if you were a smoker than if you weren't. But these are small sample sizes, so take that with a bit of pinch of salt. But we are finding higher rates in sinuses in smokers at Barton-upon-Humber. And we have the exact same kind of outcome with pulmonary inflammation. So inflammation of the lungs, which is really kind of quite heavily related to smoking. I looked at this in past skeletons, and similarly, we actually found quite similar rates in London, even a bit higher in non-smokers, weirdly. But again, at Barton-upon-Humber, higher rates in individuals who were smoking. And then I looked at limb inflammation. So this is when soft tissue in the region of the limbs becomes inflamed. It causes new bone formation on those limbs. It can be related to lots of different causes. So not necessarily diseases related to smoking, but it is also related to systemic inflammation, which is linked to smoking. And again, very similar rates in London. Much, much higher rate Barton-upon-Humber in smokers. So this is really interesting. So I was like, okay, I'm seeing high rates in Barton of inflammation. So I decided to take all my indicators of inflammation in my skeleton and grade them on the amount of inflammation someone had. So if someone had inflammation on a long bone but nothing else, then I give them a grade of 1. If they had 2 types of inflammation, say on the ribs and the long bones, then I give them a grade of 2. And then additionally, I didn't do sinusitis for this one. I should have done. I'm not sure why I didn't, but instead I did periodontal disease. So periodontal disease, we all know what gingivitis is, inflammation of the gums. So if it becomes really bad, what you get is recession of your actual underlying bone. And so I can record this and say there's probably evidence that they had quite inflamed gums. And periodontal disease is linked to lots of different conditions, but it is considered to be linked with smoking. So I took all these, I graded them 1 to 3, based on how many indicators they had. And again, St. James, London, very, very, very similar results. They're basically identical, non-smokers and smokers, but much more severe grades of inflammation in smokers from Barton. So smokers from Barton had more than 1 type of inflammation, whereas non-users normally had only about 1 type of inflammation. Does that make sense? I see some nods. And this was, statistically significant. So this is really interesting. What's going on in London that our non-smokers and our smokers have pretty much similar results, and what's going on in Barton that we don't? Well, one of the major factors is probably the influence of pollution, secondhand smoking, the urban environment, poor sanitation. So all of these factors are linked to these kind of inflammatory diseases. And it could be that what we're seeing is that in London, people were so poorly affected by the environment that it didn't actually matter whether you smoked or not. You were still likely to develop, just as likely to develop this disease. And London at this time is horrific. There is a sanitation crisis. There was the Great Stink of the Thames in 1950 something, I can't quite remember. And people were basically drinking their own sewage water. We also had the great smogs that could kill up to 1,000 people at a time. So fogs and smogs were basically a result of mass coal consumption in the city at the time. So yeah, it's not a great environment, and we're actually seeing that both non-smokers and smokers were equally poorly affected. Whereas at Barton-upon-Humber, which is a very rural site, we're not seeing we're seeing the effect of smoking. We're seeing the effect on the skeleton, which is really interesting for us because we can see that effect. It's just maybe not as simple as we thought it was. We have to understand the complexity of different factors going on at sites that are going to affect health of these people. This is just to highlight again the impact of pollution. It's impacting our biomolecular results, and it's also probably impacting our biomolecular results, and it's probably also affecting our disease prevalence rates as well. So what have I concluded from this? For now, I'm still working on it. I'm still looking at different diseases. But for now, what I'm concluding from this is that while rural and urban populations consumed in similar frequencies tobacco, the impact of the urban environment kind of obscures the consequences of tobacco use. But in rural areas, tobacco use may have had significant impact on inflammation within the body. It may have been a major contributor to poor health in less polluted areas, so in rural communities. And this is really important for anyone doing osteoarchaeological analysis of rural post-medieval populations, because smoking is not generally discussed as a factor that may have been affecting health in these populations, and actually we're seeing that there is quite a big impact from smoking on the health of these populations. I have time. I'm just going to run through really quickly. This was kind of optional slides based on how long I kind of waffled on for, but just talking about the interdisciplinary approach the project is taking. Because I think it's really interesting, the approach we have, and it kind of shows the future of these kind of big projects. So we've tried to incorporate loads of different sources of evidence. One of them being, Sarah's done some great work on historical sources and material culture. So kind of pipes people using, the types of pipes, their alternate uses. So she wrote this. I recommend anyone check it out because it's quite an odd, unusual paper, but it's about the alternative uses of clay tobacco pipes in England and Wales. And basically her and Angela Murr from History went through and found a bunch of criminal texts and medical texts about what people were doing with pipes other than smoking from them. And some of the uses are pretty disgusting. People use them as catheters. They use them to kill other people by poking them in the eyes and such. It's kind of like the weapon of choice to hand when you're sat in the tavern, because everyone's smoking a pipe. You can just jab it in someone's eye. It was used multiple different medical uses. I think one of the other ones was using to help with breastfeeding. What else was, someone tried to shoot Queen Victoria, I think, with a clay pipe and stuck inside a gun or something. I don't know. There's all kinds of odd stuff. It's a really interesting paper. But what it goes to signify is how important pipes have become within the culture of the time. Like, they were integrated in every part of people's lives, and they were to hand all the time. And they weren't just used for smoking. They were used for loads of different kinds of things. We also tried to take historical sources, material culture, and human archaeological remains, but in this book chapter that kind of hits on the stuff I spoke about tobacco consumption and social identity in industrial England. So if you're interested in the stuff I went on about identity and who was smoking, who wasn't, check out this chapter. It's literally just come out in this book, although the online free access is still not up. Yeah, I checked today just so I could be like, hey, go check it out, but it's still not up. But if you're interested, just let me know. I can send you any of these papers. So that was a nice multidisciplinary thing we were asked to do. We also been looking at dental diseases. So Sarah's looked at dental diseases in the Dutch skeletons that I haven't spoken about today. And we've applied metabolomics to loads of different things. So not only to human remains, but historical botanical remains. We actually were able to determine the provenance and the species of historical tobacco by using metabolomics. We actually found that one of the historical samples from Kew Gardens had been mislabeled as the wrong species, and we were able to tell that just by applying metabolomics. And we could say what country the species were grown in as well, the samples were grown in. So that's really interesting, very useful for anyone doing stuff on historical plant remains. We've also tried to apply metabolomics to human teeth. This had varying success, but it was on a very small sample. So we tried to look for Black Death in dentin of teeth, and we were able to find metabolites. So we do know that metabolites are hiding out not only in bone, but also in teeth. And of course, we've done all this stuff on human bone. And Sarah's applied other biomolecular techniques. So she's done calculus analysis. She's been looking for pathogen DNA and proteomics. She wants to understand how the oral microbiome changed once tobacco was introduced and people were smoking a lot of tobacco, how that affected the oral microbiome, which in turn affects your oral health. So we see people with really bad dental disease who smoke and how those factors interact together. So just to follow up on our kind of metabolomic stuff, we are the first people to ever apply metabolomics to human archaeological bone. One of the first to apply it to human bone, to be honest. Not much has been done at all. And we've shown that you can find metabolites in human bone and you can use it to understand past conditions of people in the past. This is revolutionary. Don't want to toot my own horn, but it actually is. Maybe in like 10 years or so, this is going to be I think it's going to be one of the big techniques to use on archaeological samples to understand past conditions. It's going to be massive. It's just we're just building that profile now, but once people get a hold of it, it's going to be huge. Because it's not just relevant to identifying people who consume tobacco. We can look for people who consumed other intoxicants. We can look for what kinds of diseases people had. We can tell if someone had tuberculosis, say, or not. In principle, we still need to do tests and stuff, but in principle, we can tell this stuff. We can tell what kind of environments people lived in, what lifestyles they lived, what kind of nutrition they got. We can even use it to determine sex and age. So the future applications of the method really are revolutionary. And if you want to read about it, we wrote a review on metabolomics in archaeology, which, I am actually tooting our own horn now, so yeah, that's what we've done. That's what we've found. But there's obviously still so much more to explore. So I've looked at loads of disease data. So those were just a few looking at inflammation. There's so much more. We've looked at cancers, vitamin deficiencies, dental and oral health, and more. And I need to start looking at those results and understanding what's really going on, because it's hugely complex and it's not easy to work with this kind of data. We also need to look for further differential use in other social groups. So the stuff I showed today, I wouldn't say it's truly intersectional. We weren't looking, I didn't break down those groups into, say, poor women or rich men or, all the other facets of identity. It's really difficult sample size wise to do that with the kind of samples we have. You have to have huge sample groups with known status, known ethnicity, maybe known occupation, all kinds of stuff. It doesn't mean we're not going to try, but I just haven't gone into loads of detail on that. That's definitely something that needs doing in the future. And also children. So in our Dutch collections, we know children were smoking from as young as like 5 or 7, really young. They have pipe notches that young. And we know from historical context that children were smoking really young. So we want to, I've been looking at adults, but we do want to explore how this was affecting children. Also how maternal health of smokers was affecting children born to, so children born to women who were smokers, that kind of thing. There's probably a lot we can look at in that sense. And obviously differences within and between countries, so that's why we're looking at the Netherlands. I'm sure Sarah wants to explore many other countries and populations. And also, interestingly, the impact of changing fashions in tobacco consumption. So it wasn't cool for women to smoke in public until the late 1800s, and then you get the introduction of the cigarette, and everyone realizes they have this mass market that they've totally underexploited, which is the woman. And so you have adverts like this: keep skinny by smoking cigarettes, and I wonder how those changes in fashion and the way it became more okay to smoke in public or to smoke different types. Yeah, the cigarette you can smoke much more frequently. How that affected health as well. This is us. These are our collaborators. There's probably more. I kind of lost track after I put these on things, but yeah, that's it. Thank you very much for listening.
Thank you so much. It was a really interesting
Oh,
talk.
thank you.
And this is the time for questions for Anna. So if anyone has any questions, I will pass my questions. So first over there, and then okay.
Thank you very much. That was really interesting.
Thanks.
It was particularly striking to me as a total outsider, the impact of the passive smoking that you showed, the difference between the groups in London and the more rural population. Presumably there also would have been effects of passive smoking in the rural population.
Yes.
And is there any way that you can see what that is?
Yeah.
And are there other groups where there's definitely no passive smoking? Like, are there particular social groups or religious groups that would not touch this stuff at all that you can compare it with? Or
Yes,
just interested in your thoughts on that.
Yeah, we get this question every time because it's so impossible to tell. So the biomolecular stuff might be picking up on passive smokers. We're not sure how the molecular signature works. So those unknowns that get categorized into the smoking category may have been passive smokers. We're not sure, but it's basically impossible to tell how much the impact of passive smoking had on individuals. I kind of assume that it may have had a lesser effect in Barton because people were working outside a lot of the time and the air was cleaner, possibly. So whereas in London, people working in, say, like smoky factories or offices or indoors quite a lot of the time. So they may have had a greater impact from secondhand smoking. I'm not sure, to be honest. I think it's a cumulative effect. I think secondhand smoking was happening at Barton upon Humber. I think in London, it's just that there were so many other things going on with the poor sanitation and poor air quality and that kind of thing. But it's something we're really interested in and we want to look at more. We are looking at so one of the things we did was we looked at a pre-tobacco control group from Barton upon Humber. So Barton upon Humber has burials from really early, like Anglo-Saxon, all the way through to 1855. So we took a group that were definitely pre-tobacco, so pre-1500, and we put them into our biomolecular model, and they all came back as non-smokers. So we used that as a control group to check that our biomolecular method was working. And then I've done some comparisons between health changes between the medieval period and once tobacco was introduced. But again, there are so many factors that change between those two time periods. It's very difficult to say, oh, well, there's a way higher rate of disease in the post-medieval period, so that must be because of tobacco and not other factors. But there's so many other factors. So it's really, writing papers on this is incredibly difficult, and I have to present like a lot of caveats. But yeah, secondhand smoking is something we want to expand upon and see what we can do. We're not quite sure at this point, but we're aware of the issue. Yeah.
Yeah, thanks for the talk, Anna. That was great, and I'm a bit vicariously exhausted by the amount you've done. I'm interested in your nonconformists, or like your elite woman with a pipe notch, or the elite men with pipe notches, for that matter.
Yeah.
Was wondering about if you could say a little bit more about what you make of them, or what the value of the oddballs is.
So when we wrote the book chapter on identity, we not actually really touched on this that much. And as I had to write the book chapter in a bit of a rush, so I didn't get to really expand on this as much as I wanted. But I was so interested in this idea that you have these pipe notches that certain groups have and certain groups don't. And then you have like some odd high status people who just don't care and have pipe notches anyway, or maybe they were social climbers who moved up the ranks and managed to be so well off by the end that they're buried in the very high. It's just so interesting. We have some known individuals that we've only just started from London that we started to match up with our data. I wanted to do it all blind, so I didn't know their ages and whether they're male or female, that kind of thing. But we're now adding that data in, and I'm going to cross-reference it with the pipe notches, see if we've got any high status people with pipe notches. I can look at what they did for a job, maybe how old they were, all that, the kind of things that maybe affected people's choice to smoke. Particularly, you see this association with older women and smoking, poorer older women and smoking, and I think that's really interesting as well, because it seems to be by that point they just don't care. They're like, we don't care about the fact that we shouldn't be smoking in public. They just do it anyway. And so they're written about as being old, weird ladies. But I think they just got to the point in life where they just don't care anymore. They're too old to care or whatever, So I think there's so much more that could be explored about this. And I want to do more on the identity stuff. I really do. I think it wasn't an aspect of research that we were expecting to do when we came into this, but it's turned out to be so interesting, particularly the way that the pipe was used as a kind of control over how people perceived of other social groups, which hasn't really been talked about at all by historians, or maybe it has and I've just completely missed it, but I think that's so interesting. So yeah, there's more to be done, but yeah, I think there's a few rebels out there who just didn't care and they just did what they wanted,
Do we have any other questions? So I will go deep.
Yeah, thanks. It was really interesting. You mentioned that you can track other kinds of intoxicants using this method, and I was wondering what kinds of intoxicants can you track, and also these signatures you mentioned. Do you basically have to map them onto pre-existing frameworks that people are already using? Because you sort of mentioned that you're already using these osteoarchaeological things as a background, or is there something in the signature itself that can speak about like the chemical composition?
That's a really great question. I wanted to say more about this in the talk, but I was worried about time, so now I've got a chance. Great. So there are two ways of doing metabolomics. There's one called the targeted approach. So this is when you have previous studies that have identified specific metabolites related to certain physiological processes. So say they've identified metabolites related to nicotine. And then there's another approach that's untargeted. So that's when you get a big population. This is our approach. You get a big population, you split them into two groups where you have knowns and not known. No, you have smokers, non-smokers, say. And then you look at all the metabolites that come back. And you can say, okay, these ones are specific to smokers, and we're not seeing these ones come up in non-smokers. So this gives you a wider range. So if we only looked for nicotine-related metabolites, one, taphonomy might affect whether it survives or not, so we might not find any. And two, it gives you a much smaller range of survivable metabolites that you're looking for. We want to find as many metabolites as possible because this material has been affected by time, it's degraded. So we need to give ourselves the best chance. So we did this untargeted approach. But what we found once we did the untargeted approach, and we've done this on, I think, 5 or 6 populations now, we found the same metabolites coming up every time. And they are highly significant. So we know that they're related to these groups that we've classified as smokers. So they're very, very likely to be specific metabolites formed when someone smokes. Now we've got that data, we can go out and collect new human bone samples and just look for those 44 metabolites or however many metabolites we've found that are specific to smokers. We can just do a targeted approach now. So that all that work's been done, and our next phase is to try and do a targeted approach and see if it works. I think it will work personally because the results from our studies are really strong. So yeah, if you want to go on to look at other conditions, you basically need to do the same process all over again, or you find medical studies or other studies that have already identified the metabolites you want. So UDIF is doing a lot of, you're going to do some stuff on this, aren't you? So she's working on intoxicants, not just tobacco, and metabolomics, and hopefully you'll have some cool results. But yeah, if you know what the metabolites are, you can do targeted studies and basically find whatever you want. If you don't know what the metabolites are, you have to do the approach that we've done. So it's time intensive, expensive, kind of worth it for the cool results. Yeah.
Thank you again for this talk. It's fascinating. My I guess background, I'm a history PhD here. My background actually looks at the juxtaposition between elite medicine and domestic medicine or remedies to infectious disease during the 18th century. So I ran into tobacco quite a bit.
Yeah.
And I'm sure you know that tobacco was considered to be like healthy. It was a remedy for a lot.
Yeah, it's like the panacea.
It was like the panacea for everything.
Yeah.
Teas and smoking and sucking on the leaves, you name it. So I could see why perhaps you're seeing women using it, especially since women were often responsible for medicine in the home.
Mm-hmm.
So why wouldn't they use it? It was just considered to be normal. We know it's not healthy. They didn't know that yet.
Yeah.
And I was just wondering if you've looked into like those historical avenues into why it was used by so many different types of people.
Yeah, our research is ongoing. You might like to read it actually. So this book chapter, this one from this book, we do a case study. So we found a woman from one of the populations we have in the university from Coventry, and we actually knew who she was. She's called Sarah Green. She was, I think, 57 when she died. We know her husband, etc. We know her job. She was I can't remember, some kind of nurse, basically. So someone who probably went out and visited people and helped them. She has crazy lingual staining. She was smoking quite a lot, and she had really bad osteoarthritis in her jaw. So we went through and said, well, what are the reasons she may have been smoking? One, she was older, and we see that smoking is more prevalent in the older women according to historical sources. So, one, maybe her job impacted that. Another one, maybe she was just in pain, and this was a way to try and help that. So we do explore it a little bit, and Sarah really wants to write a paper on some of the historical perceptions in the past around whether tobacco was good or bad for you, because there is some kind of counter dialogue around actually everyone's just willy-nilly using tobacco as a cure for stuff, and doctors aren't prescribing it, you shouldn't be doing it. And they were saying this in the past, but kind of got drowned out by all the people who are just, tobacco, great. So yeah, check out our chapter if you can. It should be free, but if not, just email me and I'll send you a copy. Because you might be interested.
Well, my question actually builds off of that, because I was thinking the same thing of I remember you and Sarah talking about, tobacco causes necrosis. That's why things don't hurt anymore, because it's just literally dying in your face. But besides that, is there any way with your approach to looking at it, specifically the metabolites, thank you, to see if there's any difference with life cycle? To see if people smoked and then stopped or started at a later age because of this?
Yeah, potentially. So we're working with someone who does looks at the histological sections of teeth, and the very thin layer on the outer root of the tooth is kind of laid down periodically over time. And she's actually found that how that gets laid down is disrupted by tobacco consumption. So with further research, essentially, we could maybe see these disruptions and see where people have stopped and started. It's really cool stuff. I don't know where it's going at the minute. She's doing it as she's finishing up her PhD. But yeah, there's some ways we could try and look at it. I don't think metabolomically we can, because we're still not certain the lifespan of the metabolites in the body when the person's alive. So what we have done is we've collected bone samples from cortical bone. So the cortical bone is very dense and it doesn't remodel very quickly. So the metabolites trapped within the cortical bone are likely to reflect a long period of time. We also sample from trabecular bone, so that gets remodeled quite often, and the vascular supply is a lot higher. So it probably reflects kind of what someone was doing just before they died. So we haven't analysed that data yet, but we're wondering if we see different signatures based on their cortical versus trabecular bone, because maybe they were consuming a lot just before they're dying because they were really sick, and they were trying to use it to help them. So we're looking into it. It's one of those things where it's just going to take time for us to find any answers if we ever do.
So that was our last question. So I would like to ask for everyone to do another round of applause for Anna.